Hello! In this video, we will be walking through our work on automating landmark detection in long leg radiographs using a convolutional neural network. Over the next few minutes, we'll cover the project's motivation, the dataset we acquired and prepared, the network architecture and loss function, our key results, and wrap up with a discussion of the project's limitations and future directions. Long leg radiographs, or LLRs, are the gold standard for several imaging assessments related to the lower limb. One common application is that they can provide precise views that clinicians use to characterize mechanical axis alignment during weight bearing. These images are also commonly used in pediatric practices, where clinicians use them to diagnose deformities, including bow legs, knock knees, and leg length discrepancy. Typically, clinicians manually identify several anatomical landmarks and compute angles and distances between the points, which is both time-consuming and prone to observer variability of up to 10 to 15 pixels. For this project, our goal was to build an automated method that could achieve near-human accuracy to reduce the workload placed on users who perform these measurements manually. We used 100 long leg radiographs from the publicly available Osteoarthritis Initiative, specifically found in their study 2491. We manually wanted to label 3 anatomical landmarks on each leg as ground truths, including the top of the femoral head, the medial femoral condyle, and the tibial plafond, resulting in 6 total landmarks per image. These annotations were completed manually and exported into Excel with the help of MATLAB scripts. While we treated our annotations as our ground truth, it's important to note that there are some inherent errors in the labeling. Once labeled, we pre-processed each image by resizing it to 640 pixels in height and 192 pixels in width. This allowed us to reduce memory usage while preserving anatomical clarity. We also normalized all landmark coordinates to be between 0 and 1 so that the model would be scale invariant. We then split the dataset into 70% training, 15% validation, and 15% test. This split was kept fixed throughout the project so that our final evaluation would be performed on completely unseen data.
For our model design, we developed a convolutional neural network. The input to the network is a batch of grayscale images. We then apply 4 convolutional blocks, each consisting of a convolutional layer, a ReLU activation, and a 2x2 max pooling layer. After 4 blocks, the feature map is reduced down to 40 by 12 pixels across 128 channels. This 3D feature map is then flattened into a one-dimensional vector. That vector is passed through 3 fully connected layers that reduces to 12 outputs, corresponding to the x and y coordinates of our 6 landmarks. Each of these outputs is passed through a sigmoid function to normalize the coordinates. To train the network, we use Huber loss. This loss behaves like mean squared error when the prediction is close to the ground truth, but switches to mean absolute error when the error is large. This behavior is ideal for our application as it mitigates occasional labeling outliers while still encouraging precision. We also use root mean square error, or RMSE, as our main evaluation metric. RMSE provides a direct and interpretable measure of prediction accuracy by reporting the average distance in pixels between the predicted and true landmark positions. Now let's take a look at how the model performed with training. In the top left plot, we show the Huber loss over 50 training epochs. The training loss is plotted in blue, while the validation loss is plotted in orange, and they both decrease similarly as a function of epochs. We also plotted our final Huber loss from our test set as a fixed dashed line. On the bottom left plot, we've shown the test set pixel distance accuracy for each of the 6 landmarks. The hips in red were off by about 17 pixels, while the knees in green were off by about 8 pixels, and the ankles in blue were off by about 10 pixels on average. Overall, the average landmark was off by about 12 pixels, which is comparable to human accuracy. To the right, we've included 2 evaluation samples, one with low prediction accuracy on the left, and another with high prediction accuracy on the right. The ground truth landmarks are in yellow, while the model predictions are in blue. To wrap things up, here are some key takeaways and next steps for this project. First, while our model performed relatively well, its accuracy is limited by the noise in our ground truth labels. Since these annotations were done by a single user by hand, it is likely contributing to why our accuracy couldn't get better than around 12 pixels of error. Also, our dataset was only 100 images long and came from a single study, limiting the training diversity of this set. We also tried some alternative architectures, including a heat map regression, which predicted a probability map for each landmark instead of a coordinate, but it struggled with limited training data. We also explored using a ResNet 18 backbone pre-trained on ImageNet, but that version tended to overfit unless we froze almost all the layers. Going forward, we'd like to expand the dataset, ideally including images from multiple institutions with both pediatric and adult cases. We'd also like to use annotations compiled from several clinicians to reduce ground truth noise and potentially try semi-supervised methods or pre-training on unlabeled X-rays to improve feature extraction. In the long run, we envision combining landmark detection with bone segmentation and possibly predicting uncertainty as well. With those improvements, we believe this type of model could become a useful tool for radiographic workflows and improving diagnostic consistency. This concludes our presentation. Thanks for watching.